Welcome to this talk. It's designed to educate and prepare members of our club to actually do this on these exact instruments that you could meet in the lab here in Lincoln and actually process a lot of our samples for DNA and send them off to be sequenced. And I have the equipment and supplies, which most of which I own personally and don't belong to the university, but they're there housed upstairs where I can bring them down to the lab room. And if we did that every year, it would continue us in citizen science work of preparing DNA samples and getting DNA sequence for unknown mushrooms for any floristic records and lists we're keeping, and contributions to any of the fungal diversity survey groups that we might be participating with, but also simply for identification. So we learn that one's the correct identification for those we're stumped by. And that's a great learning curve just to do that with your unknown mushroom and then come back and say, that's what it is. Okay. So over time, we ought to be able to do this at least for 10 years before I drop dead, guys. The videos will be up there for use, and the lab, according to my chairman, will be available for us to use a teaching lab at UNL when we need to, including next week or next month or sometime if we need or want to prepare some samples. And now when I present this video I've made, I don't want you to think that this is something I'm imposing on people who are just interested in photography or artwork or dyeing. It's none of that. You don't have to participate or be bothered by us who might want to do DNA work when you're part of this group. That's not what the objective is. And you may want to participate only at the level to identify something you can't figure out from your keys. And that's really the best use of this until we have a diversity survey group that are our own new iNaturalist study group to prepare a flora for. For example, if Chance wants us to work on India caves over time, we can accumulate the DNA in the sequence for his specimens, and we get to say have the one foray like we're having now, and then Chance could go as he does yearly over the year, and everything we could contribute formally to the Nebraska State Museum herbarium, where I have connections and I can actually go in and do the work for them and putting the specimens away if necessary. So I wanted to share the screen for the Fungal Diversity Survey website for a moment. Okay, if you're not familiar with this site, FUNDIS, or is everyone able to see that now?
Yep.
It's a fungal diversity survey site. And over here on the right, we have a menu that shows what projects are available. And if we look at some projects, we can find some near us that are looking for DNA sampling. But also it tells you how to get involved, and it begins talking about iNaturalist. So if one thing we should do as citizen scientists is get involved with iNaturalist, loading some of our pictures. And I've noticed Chance, for example, has loaded some of his up on iNaturalist, and some of the rest of you might have too. I haven't searched all your names. All of these can then be fed into the fungal diversity survey and into the particular project that you wanted to go into as just the pictures you took for iNaturalist and the data you wrote down there when you included that. So what they have here is all the discussion about herbarium samples and DNA and photography and submitting specimens to a herbarium. And I can't write it or explain it any better, but we can talk about this throughout the year as we get our own projects going. When I looked at projects, I looked at maps for our area, but there were only 2 markers of projects in Nebraska. But I checked those map sites in Nebraska or Kansas and Missouri nearby. And they're not actually a 4A site where you would expect them to be collecting samples and contributing to that fungal diversity project. They seem to be a whole regional spot someone's put down. And I found that the one spot that invites volunteers to join it was put down by the Western Pennsylvania NAMA group. And I've contacted them, and it turns out to be not a nice natural park or something in Nebraska like I hoped it was, but it's just a project for all of Nebraska, essentially. Why they put this spot on the border with Kansas is a little confusing also. But we can always contribute anything we put in the iNaturalist connected to that spot. But soon we'll get that coordinator. His name is John Filst, to work with us. And he has a great mycologist working as a scientific advisor for the Pennsylvania group, who I know, Walt Sturgis, and the two of them will help us on this project as we accumulate things for Nebraska and have them logged in. But we can go ahead and create our own sites. As a group, we can create one, for example, for India Caves, and we can create one for parks around Omaha or Omaha and Lincoln, or we can create one for the Kearney area. It doesn't, we're not limited that way. And anyone in the club who's using iNaturalist, if they collected it at any of those sites, could connect that to that project, and that could be the limit of it. Or we could formally prepare DNA and herbarium specimens for those sites as everyone wants us to, and put them in the Nebraska herbarium. So from this point on, I'll leave the fungal diversity and go on to talk on how to prepare the DNA samples with the instruments and methods that we would actually use in a lab at UNL East Campus as soon as we schedule it or people want to attend. So now I'll share my screen for that talk. Hello, the plan today is to try filming and recording audio as well on the computer next door while I demonstrate how we save DNA from a mushroom for all time and preservation in a herbarium for identification. First, we have to put on gloves because this is not dangerous to you, but your hands are covered with DNases, which destroy the DNA that we're trying to preserve on the cards. And we take our mushroom, and we're going to section a part of it and smash it onto our card with this hammer. Now the cards are called Walkman plant cards. But the Walkman, oops, this is the wrong. Walkman plant cards. The FTA is the particular type of membrane of cellulose on these squares that captures the DNA
from the crushed cells of any organism. From that point, the DNA is protected for over 20 years, they claim, at room temperature, simply as a dried card in an envelope. And they do supply a sealed envelope. Now, one problem is when you squash your mushroom or plant or animal on there, you get juice on there, so the card is not really dry. So one thing we do is also buy little desiccation packets sold by Walkman for these particular cards and throw one in before we seal the envelope. Now the reason there are 4 squares is for replication, scientific replication. And this will allow you to send 1 square off to a DNA sequencing facility company, and they can extract the DNA and process it and give you your phylogenetically informative DNA sequence to identify your mushroom for about $30 today, and I assume in 10 years it'll be down to $15. Now, we could extract it ourselves from the card in the laboratory and then send it off for about a $10 charge for the sequence. But you have to put in your labor and time. But regardless, you have 3 extras there in case anything goes wrong, or to deposit with the herbarium specimen in the Nebraska State Museum herbarium, for example, with your dried mushroom sample. So I've used this in the field in Alaska for diseases of trees, not just simple things like mushrooms, but removing the bark of a tree, finding the diseased cambium, and crushing the diseased cambium onto those Walkman cards, and then processing it and getting the DNA of the pathogen as well as other saprophytes and endophytic fungi from those cards. So let's begin with the typical grocery store button mushroom, Agaricus bisporus. And we know from biology that the area that has the most activity for DNA replication is the sporulating area, because every day and every night this mushroom, which is still alive, is producing new spores and therefore replicating cells and producing new DNA copies. So we at best smash the part of the mushroom that we call the gill, or perhaps the pores if it's a polypore. Using our gloved hands, we take the card and we don't touch those squares. We probably need half of this quantity, so I'm going to trim it to half. There may still be too much juice for it. Yes, that will be too much. So let me trim it to about less than a square centimeter of gills. Now you can, of course, first squash it with your finger. And the purpose of the hammer is really to break cell structures, and since this is a plant card, some of the instructions we get are for more rigid cell walls and the fungal cell wall. But from experience, I've learned that this works very well on everything I've used it on, including wood. Not the dead part of wood, but the cambium. And you could hammer until the paper becomes a little fragile and decide to stop, or you could stop about where I did there. If you look at it, it's very compressed onto that 1 square. Now, you would repeat this 3 more times in order to have replicates of this 1 mushroom. And then you can write any information you want on the card, which I highly recommend you do. Now you can write it on the envelope as well, and we put the card in. So we would want to insert a desiccator packet, or maybe 2, because that's kind of moist, and we want this to last forever. So we put it in there, remove the automatic sticky surface sealing, and we seal it and write your information on the cover using a permanent marker called a Sharpie or something along those lines. Now you can at any time refrigerate this for extra security, and it may last 100 or more years. But all we know is that the guarantee they give us is up to 10 years at room temperature. Now this makes sending samples very easy in envelopes through the mail. I received the whole packet just a few days ago for scientific studies. Now there's a solution that is sold with these cards. The cards were developed for red blood cells in human blood serum for extracting DNA. Even though this says plant card, it was developed for the medical people and used for blood work. And they found a particular solution helped to extract the DNA from this most effectively in a single step or two. And then they would send it off to the sequencing facility as extracted DNA rather than a piece of card. Or they could amplify it, as I'll show you in a moment, using polymerase chain reaction, PCR, which we'll talk about in our next film. So a small hammer, sealed envelope, dry right packet, hopefully. And these are sold in boxes of 100, so you do have to put in an investment to begin with. This is $100 worth, and it's 100 cards. Now to prepare or extract the DNA from your squashed gills on that FTA card, this is a solution they recommend, and they send you a small recipe and a booklet on how to use this. This is just called the FTA purification reagent, and for 100 mils by Walkman, this is probably a lifetime supply even for my lab. You don't have to autoclave it or anything, it's ready for use. And if it works that way, that's the fastest and easiest way to extract your DNA from a mushroom in any manner that I know of. Plus, if it fails, you still have your permanent, 3 more of your permanent store of DNA on the FTA card. And you can try another method, which I've done before. So if the DNA is particularly nasty with compounds like a hemlock tree I was working with, when I was trying to preserve the pathogens in the fresh cambium of a hemlock tree, they're loaded with toxic compounds that made extraction of DNA difficult from the card. So the standard procedure is to use a very well-known solution present in every lab that does DNA work, referred to as TE. It's a buffer called tris at pH of 8, and it contains the chelator, EDTA, like you use for plant growth. So why? Tris is set at pH 8 because the DNases that eat up DNA cannot work at pH 8. The EDTA chelator is present to grab all the magnesium from the specimen because DNase requires the cofactor magnesium present in the solution. So simply by using TE to dissolve the DNA out of your cut strips of FTA card in a little plastic tube, not only protects it from DNases, but removes it from the cellulose substrate sufficiently to work with the DNA. In any other method, the DNA is purified and sent off to sequencing facilities, whether they're commercial kits or simple chemical compounds. We're talking about DNA extraction with kits or chemicals, And they do the same thing, so I'll go through the typical procedure very briefly for you. Now we have our gloves on to protect our DNA throughout the whole procedure from our own DNases. Now in the kit there's usually 3 to 4 solutions, and the last solution that they give you is basically TE that we mentioned before, that preserves the final purified DNA. But today the DNA is so pure out of these kits that we use as a substitute just pure water. Only this high grade of water though. So in solution 1, we basically have the typical tube that we just ground the DNA in with the small blue mortar. Pestle, the mortar is the tube and the pestle is the blue. And we've incubated it for about 30 minutes to 65. Now, that means most of the cells have burst in that solution with this detergent SDS or SLS. Now we centrifuge this for enough minutes, usually 5, to knock all the debris down from the cell walls and burst cell membranes and such down to a pellet. And then we essentially pour the top solution into a new tube, and we throw away that pellet. So that's got rid of the cellular debris. Now we have a solution that contains DNA, proteins, carbohydrates, lipids, fats, all the things that are in a burst animal or fungal cell. Now we have to knock down and get rid of everything but the DNA, or it will interfere with our future work downstream. So the next solution, so that was solution A that we burst the cells with. Solution B, whether it's in a chemical kit or your own solutions, now that will be added to knock down the carbohydrates and some of the lipids usually. So you add B, you may incubate it briefly, put it on the centrifuge again, or we usually mix it for best results on a vortex, which sort of creates a cyclone of liquid inside. But you can get away without an expensive instrument, although this was surplus and it's worked for me for years. It's just to tap it with your finger, snap it, and you mix the solution back and forth and dissolve anything that might have needed dissolving or mixing. And you put it in your centrifuge, and you centrifuge it for about 5 minutes more. And then what your pellet is your carbohydrates and some of the lipids you were trying to get rid of, because of the nature of B. So again, if you have a good strong pellet, you just pour off the liquid into a new tube and throw away that pellet. At this point, your DNA is getting purer and purer. You've gotten rid of the proteins and the cellular debris. You've got rid of carbohydrates, polysaccharides, lipids, fats. Now what we do, and every kit is like this, while you can't do this with your chemical procedure, the kits have this one secret that's shared by almost all kits. They sell you what's called a column. It's really just an internal holder of a membrane that fits inside the 1.5 mil tube. Now those membranes, they're all secret formulas, but in reality what they're doing is it's essentially a glass wool, like fiberglass insulation. It's charged positively, and we put that into a new tube. And we take our clean solution, that our DNA that was left from after step B, that should be rid of many contaminants, and we pour it into that little chamber above the filter membrane. Then we add a certain chemical C, and that's to change the pH and condition things so that the DNA gets stuck on that glass wool when it's centrifuged. And all the liquid that comes out the bottom is just extra the salts and chemicals and things that are another type of contaminant. So everything's above that membrane when you put it in a centrifuge. Everything's below that membrane after centrifuging, and your DNA is stuck as negatively charged DNA. Is stuck on that positively charged glass wool-like membrane in this column. Instead of throwing out a pellet, you're throwing out the solution, you're getting a clean tube. Now all your DNA is in there. There's no liquid above, and there's no liquid in the new tube. Now you use solution. You may wash with solution C twice, just to make sure that all contaminants are washed through, and that means you just centrifuge it, throw out the liquid, centrifuge it again, throw out the liquid. Now, you want to release your DNA, so you always use either a new tube, you use a new tube at that step, no matter what. So your DNA is all bound up in that little white material. Can you see that? So of course, solution D is chemically designed to release it and drop it out of the glass wool and into your tube. And it turns out pure water will do that, or TE will do that. So your last solution is generally TE, or pure water. You fill up the top of the column, you spin it in a centrifuge, and everything that washes through is all your DNA released into those solutions. Now that's very pure DNA at that point, and we don't have to worry much about its safety and handling anymore. But for conventional purposes, we throw away the column. For conventional purposes, we keep it on ice or at minus 65 to keep in the good habits. So here is more or less 2 drops, maybe normal drops of your containing all your DNA from that mushroom you ground up. And it's actually too concentrated for us to use reasonably, and you'll see that in a moment. So we have to now get the magical chemicals that are used for the polymerase chain reaction, which is this instrument over here that produces millions of copies of the DNA molecule you want. Well, here we have the whole genome of the fungus in a little drop of water. What do we want 1 million copies of? Now, we can't yet work with 1 copy, except some special techniques I won't go through now that are brand new. So with normal sequencing, we need 1 million or so copies of clean single gene that you want to sequence. So what gene do we choose? Well, it turns out with fungi, we use a gene for a very specific purpose. It was one of the first ones used, and it's become a barcoding sequence. The one DNA sequence, that one gene, is all you need to identify most fungi. And that's published as the barcoding sequence. And it's abbreviated capital I, capital T, capital S, ITS. And that's how everyone refers to it, but it has a complicated name, internal transcribe spacer. Well, that sounds complicated, but it's actually a very simple idea. It's inside, it's these clear bars linking the ribosomal small nuclear gene that all eukaryotes have, and all prokaryotes and mitochondrial DNA and chloroplast DNA also have it, but theirs are not linked. The ITS links in eukaryotes this small ribosomal RNA or DNA here to the large ribosomal RNA. They function after they've been read into RNA, but the gene code on the DNA is DNA. So it turned out these are highly conserved molecules that all life cannot survive without them functioning, because ribosomes read your messenger RNA and they produce proteins. Well, it turns out that the ribosomes that do that are not one molecule. They're a sandwich of two, the small ribosome and the large ribosome. And the messenger RNA, which is the same thing that you got in the Pfizer COVID vaccine as injected in your arm, is read like all other messenger RNA molecules between the sandwich of ribosome, of the ribosome, and the protein is made after reading the sequence of that ribosome, of that messenger RNA, I'm sorry, with the ribosome. So what happens is the messenger RNA is the alphabet in the sentence. The ribosome sits on the messenger RNA, which was given to you in a vaccine. It reads that messenger RNA, and as it does, every three messages on there, nucleotides, like ATT or CCG, they code for a particular amino acid like lysine or arginine. And your transfer RNA, which is a small separate molecule on other chromosomes in all life, are the ones that attach the amino acid onto the three coding sequence on the messenger RNA. And it just chugs along like an engine. Every three nucleotides that are read on the messenger RNA, a transfer RNA reads and puts on an amino acid, and it drops away. So you're building a protein, amino acid by amino acid. In the COVID vaccine by Pfizer, you're building the spike proteins on the outside of the virus particle, which is what our immune system first recognizes as foreign and mounts an antibiotic response to. In this case, if we change any of the important coding nucleotides in the small ribosome or the large ribosome, it won't function and you die. So they're what we call conserved molecules. They do change over time, but they're either neutral changes that don't affect natural selection, or the positive selections that changes that positively select natural selection. And all the negative ones basically kill the organism. So you can use the sequence of this molecule, and it was the first one used, but it only has the resolution of the family. So it might tell you that your fungus is a cup fungus, but nothing more. And this one is much the same. Now, in between is an area that does not code for a functional product. It turns out that the ITS, the internal transcribed spacer, is essentially that. It's a region of space separating it, and it acts like a physical hinge, where it's just a shape, a loop-like shape that has no serious constraints on the shape of that loop. It just has to work as a hinge. So you can see a very sloppy loop or a very tight loop can all work as a hinge equally. So when we talk about the molecular clock, which we will now, we're talking about random mutations that occur on Earth in the genes of microorganisms and humans and such over time. So that's usually due to UV light or radiation in the background or accidents in your copying of the DNA. So we have a calculated rate, which we're always refining, which is the natural mutation rate occurring at all times on Earth for DNA sequences. And it turns out that since this is a hinge and it doesn't code for anything important, all the mutations accumulate there, and none of them are negative, none of them are positive, and none of them are anything except neutral. So they just load up that sequence. They don't mess up the microorganism or the human from functioning as they would in this molecule or this molecule. So when we look at Agaricus bisporus, a commercial strain, which is genetically different than the field Agaricus campestris and its natural population genetics. But they derive from each other. The bisporus is derived from the campestris in the wild, and it occurred quite a long time ago as a change. And so they are different species, but the branch, the amount of changes in the ITS are rather minor because they're the closest possible brother and sister species. They just separated from one another, perhaps in the time that humans have been cultivating them. So it's recent changes. But if you look at the population of campestris, the DNA sequence that you would read in that ITS would vary maybe 10% from the DNA sequence of the bisporus that is used for cultivation versus the wild population. Now, if you're using clones, of course, of a bisporus cultivar that you particularly like, a brown version that stores well and tastes good, there'd be no changes year after year as you clone it and use it. And or through reproduction either, there would be no changes. But the campestris population in the wild is always slowly changing with the molecular clock and selection, other things, genetic drift and such. So the size of this molecule is 600 base pairs. It turns out most of our sequencing instruments, they can't read more than 800 base pairs accurately today. So it's a very good size for getting accurate DNA sequence of and paying one price. If we did this, which is 1800 base pairs, we'd have to send it in as small pieces, maybe 4 pieces, to get an accumulated accurate molecule. And this one, which is 2800 base pairs, the large ribosome, it would take 6-7 molecules to try to cover that accurately. But we can often get the entire ITS with a single tube sent to the sequencing facility for one read across, adequately enough to identify the mushroom in our hand. Now, the reason this is up here, it's not that my students don't understand that, it's these little codes on top and these little codes on bottom. Let's see if you can see that. They're what we call abbreviations for the primers. A primer is 20 base pairs, a little fragment on that molecule. So we have primers all up and down these molecules, and we use them to make copies between the top primers versus the lower primers. Because the top, while it's a cartoon we draw, it's actually represents the top strand of a double-stranded DNA molecule, and it's read from left to right on top. And life on Earth reads the bottom as right to left. So the primers up here also function from the left to travel to the right and make copies of the DNA. And then the one on the bottom is traveling from the right to the left, making copies of the DNA. So when we want a double-stranded copy of the ITS, we have some universal primers developed by friends who are actually mycologists, and they developed them to work for all eukaryotes about 1987. The ITS-1 is up here, and it's right on the edge of the ITS. And the ITS-4 is down here, right on the beginning of the bottom strand of the DNA, or the end of the top strand. So if we want to make copies of this ITS, we put in a little bit of ITS from this strand going left to right, ITS-1, and a little bit of ITS-4 from this strand going right to left, and we put in the solution, the nucleotides A, T, G, C, and we put in a buffer, and then we put in the molecule that copies DNA called a polymerase. All molecules that copy DNA are called polymerase, but the one we use for making a million copies is called TAQ, T-A-Q polymerase. TAQ stands for the bacterium Thermus aquaphilus. So T is the beginning of the genus and AQ is the beginning of the species name, TAQ. So this is a bacterium that we've discovered in the hot springs of Yellowstone National Park, where organisms were living very close to boiling and doing quite well. And we said, well, how can they make DNA and make proteins? Well, they had to evolve a polymerase especially capable of working at those temperatures. Now, I remember I told you to protect DNA, we put it at 65. Well, it turns out that this TAQ enzyme works best at 72. And so our DNA is protected all the time that the polymerase is making copies of it. That's one important step. Now, the other steps I'll describe now. In our solution with the nucleotides ATGC, a buffer, a polymerase, TAQ polymerase, and a tiny bit of our DNA, and I do mean tiny, we put in the PCR machine. Now, let me mention that to you now. So here's our tube of purified DNA. Actually, it's on an ice bucket, ready to go. You could have froze it overnight or come back 10 months later and pulled it out of the freezer, thawed it out on 65, put it on the ice and ready to go. So here we have 2 drops of DNA. Is that enough? Well, it turns out we take our tiniest pipette, and we take the tiniest possible tip, and we set it at the absolute minimum of accurate pipetting, and we go in there and we pipette out 1 microliter. We can't, with these tools or any tools I know of, accurately pipette less than 1 microliter of liquid. In fact, I always look at the tip because it's just the tiniest amount in that tip that you can see. You take that in, you're putting that in your tube of nucleotides and polymerase and buffer, and that's all the DNA you put in there. So the DNA that you extracted will last for 1,000 reactions. No, it actually will last for 25 reactions. So you have 1 microliter of your DNA in there, and solutions I mentioned before, and the whole volume is only 25 microliters now total. and we put that on the PCR machine. We, of course, always use a permanent marker and label the top of a tube with the name of the organism, so we don't forget it, or we don't mix it up with the other 19 tubes, because we could be doing 19 mushrooms in my machine now, plus 20th one. So let's say this is the chances truffle. We put CT, why not? And we got that on a PCR machine making many, many copies. So basically that's it. But then we have some magic tools. And these magic tools really help us out. What we have instead of the universal primer, ITS-1, which will work with my DNA and plant DNA and nematode DNA and aphid DNA or anything, and ITS-4, which will work with all those organisms too, to make their ITS. You ever want to make your own ITS? Go ahead. Anyway, we use an ITS-1F designed specifically to be selective for fungi. So if our mushroom was a little moldy, had bacteria in it, you know, some nematodes were crawling around, it had some molds that are invading the stalk, and it was in bad shape, we could still get the fungi out of there, all of the fungi. So if there is a mold, you would get the mold in your mushroom. But we have another magic weapon in our arsenal. We have an ITS-4B for basidiomycete. So the B, the basidiomycetes are all the mushrooms with gills and pores. It would get all the fleshy mushrooms you know, except the morels, which are ascomycetes. So the ITS-1F to ITS-4 would get all fungi, including the morels. But the ITS-1F and the ITS-4B would get only the boletes and basidiomycetes, all the gill agarics and polypores, but not morels, and not any of the molds. So by using a 20 base pair DNA sequence that's single-stranded called ITS-1F, and another 20 base pair single-stranded sequence of DNA called ITS-4B, we're guaranteed to, from all the million genes, actually it's about 30,000 genes and other junk DNA in that fungal genome, you're going to only copy the ITS, and you're going to only copy it for the DNA of the mushroom. Now this has worked very well for me in plant pathology, because I could grind up moldy or dying tissues of a plant and find out all the fungi in it. And also go to the basidiomycete primer and find out all the basidiomycetes compared to the ascomycetes in that diseased branch of a tree. And not have to worry about separating out the plant DNA, the bacterial DNA, and the DNA I didn't want. So I'll put this back up, or my students will be upset at me. And you know, we're talking about one big droplet in this tube giving you your million copies. So how does that work? This little machine I bought in 2000, it's already 20 years old. It's one of the cheaper, smaller versions of a PCR machine. So we already have the first magic, which is the Taq polymerase from the hot springs bacterium. We have the other ingredients we need to make DNA and the right primers. The primers are made for you by a sequencing company, and you get a droplet of them, and they last you 4 years sometime. So you put your tube in here, and then what happens? So we put the chance truffle tube in there. We close the lid, because the lid is a heated special lid. And you have programmed certain steps in here, but all PCR are essentially the same steps for all work done in the lab. And this is what happens. You've got double-stranded DNA in there from your original purification, and you heat it up to 95 degrees centigrade. 100 is boiling, and this is hot enough, but below boiling. It's hot enough to make all the double strands open up and become single-stranded DNA. Why do we do that? Because in the body of human body and other organisms, to make copies of genes, the double strand has to open up and let the polymerase or whatever they're using, the ribosomes, get settled down on that DNA. It would be a polymerase, not a ribosome, and make a copy of the gene you want. So we open them up. They're all single strands. So the first step here is 5 minutes at 95 degrees centigrade, and it's just making all the DNA in there single-stranded. Then from the 94, we start a 35 cycle repeat of the 3 steps. And each cycle is part of the chain reaction of making more and more and more copies, doubling what you have in your tube. So despite the fact we put 1 microliter in that tube, we probably had 1,000 copies of the ITS in that 1 microliter. And now they're all single strands. And so the first step is from now on, the next 35 steps, they go 95 degrees, about 1 minute, and make sure everything's single strands. Then it drops to about 50. And that is just the right temperature for the 20 base pair primers to attach to the exact sequence they are a complement of on the single-stranded strain and firmly anchor themselves. So the ITS-1F is anchoring itself on one single-stranded area, and the ITS-4B is anchoring itself on the other strand. Now we raise the temperature to 72, and that's only about 1.5 minutes. So what happens at 72? That Taq polymerase just goes crazy, making copies and copies and copies and copies between the ITS-1F and the ITS-4, going in both directions. So you have your tube with every cycle of the 35 cycles, making more and more copies of those 1,000 original copies of the ITS. And they're making them without primers we put in there. So if you had changed primers, you get a different molecular sequence. And in my lab, we use other DNA sequences, and their maps are up here for the student, and there too. So we're making 1,000,000 strands. At the end of 35 cycles of going from 94 to 50 to 72, 94 to 50 to 72, we stop and give it about 8 minutes at 72 degrees. Why? Well, we've got 1,000,000 or more strands in that little droplet of the ITS sequence. And we want to make sure they're all complete, that the chemical reaction has had enough time to go from the left to the right side all the way. And in the other case with ITS-4B, from the right to the left all the way to the end. So you have to complete your polymerase copies. That's just what that step is doing. And then you're essentially all done, and you put it on ice. And these instruments actually go down to 5 degrees centigrade, which is your refrigerator temperature, not your freezer temperature. And that holds it so you can sleep overnight, come back the next day, and pull out your DNA without any damage to it, because it's been sitting essentially on ice all night. At that point, your tube contains at least 1,000,000 copies of this ITS sequence. Now, what do we do with it? All we got is an imaginary molecule in there. If we're in a hurry and want to skip a day of work and throw things at the sequencing facility, we gamble, and we take part of that liquid in the tube, and we put it in a new tube according to the directions they give you I've got several written here by different companies. They want about 12 microliters, water, and your DNA and primer. That's all they're asking for. So I put in a small portion of the million copies. I put in a little water, and I put in a tiny bit of primer ITS1F. Send it off to the sequencing company, and I'll get the DNA sequence, the ATGs and Cs from the ITS1F down that molecule. It'll be going left to right. If I have extra money, or I want to publish and be particularly precise, knowing my sequence is perfect, I'll do the same thing with another tube, but I'll put in ITS4B and send it to the company. And then they give me the exact same sequence, but they're giving it backwards to me, from the ITS4 right side to the left side. Then on your computer, when you get these sequences, you flip one and you compare two side by side. And you check to make sure they're identical. And if they're not identical, if the mistakes you think are possibly there on the left side, you accept the top strand from the ITS1F primer as the accurate one and ignore what's happening with the ITS4 primer, because it's running out of steam. It's getting inaccurate the longer it goes down that strand. Same thing with the ITS1F. It's accurate near the left side, and as it runs to the right side, it gets less and less accurate. So if the mistakes or the question marks I have about the exact sequence from the pair I'm matching, if the mistake is on the right side, I choose the ITS4B sequence as the accurate one and ignore the difference that's in the ITS1F sequence. So we know that, if you use the left primer, your sequence is most accurate at the left side. You use your right primer, the sequence is most accurate on the right side. They're both the same in the middle, and you're just correcting on the computer or by eye, the accuracy of your sequence. Once you've done it in both directions, it's usually considered high enough quality to publish and to upload to GenBank. But to identify fungus, you only need it in one direction. You don't need exact accuracy. So you want to save money, you just put in the ITS1F primers in your DNA samples that you send off to the sequencing facility. You're saving half the cost. You're getting only the top strand going left to right. Well, when you blast that in GenBank, it's going to give you the right species. Now, if you only sent them the bottom primer, ITS4B, and the sequence is backwards, basically, from what we're used to, GenBank has no trouble with that. It takes that sequence and identifies the mushroom for you, just as though it was the top strand. It makes no difference. The software is designed to handle top or bottom strand and give a perfect match with what's in GenBank. So as long as Agaricus bisporus is in GenBank, we're going to have a 99% match with one run of a tube, so $5. Of course, you've done some work. So you're lazy, $30. Right? And let me tell you, if you, for example, if I was a forest scientist in USDA, I think they'd pay for that $30 and give me 2 days of work free. And I could just work with the data when it came back from the sequencing facility. So that's what it's all about. The PCR machine makes 1 million copies. The magic was using that Thermus aquaticus polymerase from the hot springs, TAC. It's only important because of the cycles we use to open up the DNA, settle the primers down on the single strands, and then do the polymerization and protect the DNA from being degraded the whole time. So your tube of 1 million copies of sequence, of DNA of the ITS region, well, it's good for hundreds of years of frozen, minus 80 the last 100 years, frozen at minus 20. Your standard freezer compartment lasts your lifetime, no trouble. And that's what my freezer compartment's all full of, a lifetime of DNA in tiny little tubes, the small ones. Oops, this is a small one. So that's all that we need to know about that. Now, other organisms in GenBank have a different barcode sequence. They don't use the ITS as their primary molecule for identifying them. All animals, including the insects and nematodes, we use a mitochondrial gene called cytochrome oxidase subunit 1, or COX-1, COX-1. It's usually published CO1 in the capital letters. That's their molecule called the barcoding molecule for identifying an animal species or nematode species. So this works for almost all organisms. For fungi, ITS works for most all of them. It doesn't work for fusariums, one of our common pathogens. Then we have to, it's evolving so quickly with crop plants in agriculture that we have to use multiple genes. You see lots of published papers with multiple genes, and it seems impossible for you to identify a fungus because they're using 5 different gene sequences. Well, that's just to break it down where we want to be absolutely certain we're right. And usually from those papers, you can find one molecule and one sequence that you can use for an absolute identification, based on that paper, in fact. So you're not usually using more than one gene sequence. So it's all the same procedure. The only difference would be the primer, little primers would be different, and you just buy those. So no problem. You're a scientist on your way to doing molecular biology.
I hope these videos will be posted. I actually did this series with the iPhone, just on a stand. And I did do it with a video camera, which is better, but it took 82 minutes to go through everything in a little better detail. And that's the one we could post in different pieces so that people can look at them in the future on our website. And then when we meet at least once a year in a lab and do some of this actual work for fun, but you know, we can have a good time and do some of this and get some ideas of things we can't do without books and keys well, and contribute. And next time we meet, I can tell you about some real science that's already come our way with people want to cooperate with Chance and truffles he's found in Indian caves. And I'll do the physical work, but there's some real scientists, real mycologists that want to figure out if those truffles are mycorrhizal or not. And they're using what's called stable isotopes, very advanced new system to look at carbon and nitrogen sources and their natural isotopes in nature to determine if they're coming from a photosynthetic plant or from inorganic minerals in the soil. So hopefully we'll get a little Zoom discussion with those scientists once they send us some work to do and we have something to contribute. But I think as a club, we can easily follow their directions to send a few tubes of material to them as soon as we can re-encounter those mushrooms. And hopefully Chance will know when they're popping up again. So till next time, it's I'm signing out.